All right, so now that we have all of our libraries installed, we now can move forward with actually downloading this data from the GEO database. And so even though we installed these specific libraries, now we have to kind of load them into our environment. And so we do that by calling this library command and then saying we want to load the GEO query library. Once we do that, it'll allow us to use this particular function called getGEO subfiles, and I want us to enter GSE8823 in quotations. And this will take a little bit of time because basically it's going to consult the database, and we'll actually come over here, way up at the top. Oh, this is the wrong one. What's going on? All right. So way up at the top here, this will consult the Gene Expression Omnibus database. And we can actually go here. So this is a one of those databases that is stored within NCBI that we've been working with for sequencing data. So because this microarray and sequencing data is kind of all in the same boat, we consult this database here, and we can look at the specific study that it came from. So it's from August 1st, 2008. And here is the particular paper that I was talking about. If you're interested in learning more about this data set, overexpression of the apoptotic cell removal receptor MERTK in alveolar macrophages of cigarette smokers. But basically, we load that in. It's going to take a little bit of time, like I said, and then we're going to use this untar command, which basically the way we download it is in a particular format, and what we're going to do is we're going to decompress that data so we can use it and load it into our system. So once we do that, we load a few other libraries that we're going to use in the analysis of this data, and then we take our raw files, being these .cel.gz files, and we're going to say, okay, load these all in, and they're gzipped. So we're going to set that to true, and we're going to call this object raw data. So remember, we create an object by using the arrow symbol and the hyphen, and that kind of produces an arrow that says, okay, read these CEL files from the CEL file that we just created into an object called raw.data. And below you should see the, once you press the run current chunk button, it'll start to work through that particular data. And so let's do that now. Oh. And see, so it says that we have an error here. It says could not find function read.cel files. And so let's go back and load our libraries. Looks like we didn't do that. And once we do that, now we have that function defined, and now it knows what to do. So it starts reading in our raw data. It's just chugging along here. And we can get data from, any, particular data file that we've defined. So, right, we defined CEL files, and we can see, okay, what is actually in there, and we can see that it's a list of all of these different files here. We can also do something similar. So let's say we wanted to insert another R command, and we wanted to see what was in our raw.data object. So we go to raw.data, and we see that it's an expression feature set, has some assay data, some protocol data, and so forth. And so now what we're going to do is we're going to take information from the paper and say, okay, this pdata command on our raw data, we're going to create a column so that that dollar symbol stands for a column in a data frame, and we're going to create a column called status. And then we're going to add to that column, we're going to say, okay, the first 11 entries in our data set are non-smokers, and the second 13 in our data set are smokers. And so when we run that, now we have, see, first, and you can go first 11 non-smokers, and the next up to 24 are going to be smokers. All right. And now moving on, so remember during our RNA-seq based lecture, we kind of discussed a bit about this, the importance of this concept of normalization. And so basically correcting for inherent biases in our data to be able to compare them between specific groups. And so let's create a box plot of all of our, so here our data is raw probe intensities that we're using to quantify gene expression. In RNA-seq, for example, it would be the counts of those sequencing reads. And so it's just a little bit different, but we're still able to quantify the gene expression from our data set. And you see that for the most part, you know, these probe intensities are centered around, you know, that 6 to 8 mark, but we have a couple that are a bit elevated compared to the others around it. And so this is what we want to correct for when we're trying to make a valid statistical comparison. And so here we can create our GSE 8823.RMA object, and we're going to perform RMA, which is a type of normalization, on our raw data. So we could run that. So come up here to run the current chunk, and if it's green, it means it's kind of running that data through your system, and it'll tell you specific points that it's hit within the analysis itself. All right, so now when we do the same box plot but on the normalized expression, we now see that it's much more uniform and ready for comparison. All right. And so now we need to kind of set up our analysis, or set up, you know, our, you know, what we're considering one group and what we're considering the other group. And so our two groups, just as a reminder, are non-smokers and smokers, and we're comparing the gene expression between those two specific groups. And so you don't need to know a lot about what this specific line of code within R does, just to know that it assigns our specific smokers to a specific group. So you see when we run this, we get our first 11, which were our non-smokers in group 1, in one group with ones in that column, and then numbers 12 to 24 in another group. And so now we have two specific groups that are defined, and this particular command here uses a form of regression to determine if the differences between these groups are statistically significant or not. And so we run that. It runs quite quickly. This is just another type of comparison that it's doing. And now we can start to get into specific results. And so let's see what our smoking results object gives us. So we've used one of those statistical comparisons that we did between our smokers and non-smokers. So you can see that our variable here is smoking or not. The adjustment method we did, this stands for Bonferroni-Hochberg, which is a type of statistical adjustment. And so when we're doing hundreds of or thousands of statistical tests at once, we need to correct for that in some fashion. And so this adjustment method helps us do that. And so now let's see smoking results. And another little tip when you're moving forward, when you start typing, like let's say smoking, so our studio will try to kind of auto-complete what you're trying to type. And so you see that smoking results is the only one. I can hit enter, or I can press tab, and it'll complete that typing for me, and I don't have to write the whole thing out every single time. So let's see what we have. So see, we've entered in smoking results and we've ran that chunk, and now we have, you know, a number of those fluorescent probes that we measured the intensity to tell us the gene expression in each particular sample. We have what's termed log fold change, average expression, and then we have p-value, our adjusted p-value with Bonferroni, and some other values. And if you want to know more about each one of these specific values, you can learn more by going to the lecture that was uploaded on Friday. All right. And before we finish up the exercise, I'll stop here and let you get caught up if you need to enter in specific types of code, or are having some issues with any of the code blocks that were entered before.